Hello friends, today we will discuss about cytoskeleton and cell movement, and our main objectives will be to give a brief introduction about cytoskeleton and its role, to study the structure and organization of actin filaments, to study the association of actin filaments with plasma membrane, to understand the role of actin and myosin in cell movement. So first and foremost, an introduction about the cytoskeleton and its role. The ability of eukaryotic cells to adopt a variety of shapes and to carry out coordinated and directed movements depends on a complex network of proteins that extends throughout the cytoplasm. This network is called the cytoskeleton. Although, unlike a skeleton made of bone, it is a highly dynamic structure that reorganizes continuously as the cell changes shape, divides, and responds to its environment. The diverse activities of the cytoskeleton depends on three types of protein filaments: actin filaments, microtubules, and intermediate filaments. Each type of filament is formed from a different protein subunit. Actin for actin filaments, tubulin for microtubules, and a family of related fibrous proteins such as vimentin or lamin for intermediate filaments. Some of these accessory proteins link filaments to one another or to other cell components such as the plasma membrane. Now we will discuss in brief the role of cytoskeleton. Cytoskeleton is a dynamic scaffold providing structural support that can determine the shape of the cell and resist forces that tend to deform it. It acts as an internal framework responsible for positioning the various organelles within the interior of the cell. It acts as a network of tracks that direct the movements of materials and organelles within cells. It is a force-generating apparatus that moves cells from one place to another. Single-celled organisms move either by crawling over the surface of a solid substratum or by propelling themselves through their aqueous environment with the aid of specialized microtubule containing locomotor organelles, cilia, and flagella. Multicellular animals have a variety of cells that are capable of independent locomotion. An essential component of the cell's division machinery, cytoskeletal elements made up the apparatus responsible for separating the chromosomes during mitosis and meiosis and for splitting parent cells into two daughter cells during cytokinesis. Coming to our next objective, that is to study the structure and organization of actin filaments. First, isolated from muscle cells, actin constitutes approximately 20% of the total cell protein. Two strands of actin molecules polymerize to form actin filaments. Actin filaments are thin, flexible fibers approximately 7 nanometer in diameter and up to several micrometers in length. Within the cell, actin filaments are organized into higher order structures forming bundle or three-dimensional networks. The assembly and disassembly of actin filaments, their cross-linking into bundles and networks, and their association with other cell structures such as the plasma membrane are regulated by a variety of actin binding proteins which are critical components of the actin cytoskeleton. Actin filaments are particularly abundant beneath the plasma membrane, where they form a network that provides mechanical support, determines cell shape, and allows movement of the cell surface, thereby enabling cells to migrate engulf particles and divide. Actin, the major constituent of the thin filaments, exists in two forms: G-actin, which is a 42 kilodalton monomer, and F-actin. F-actin is formed by polymerization of G-actin and resembles the thin filaments found in muscles. Along the actin are myosin binding sites where myosin can form chemical bonds with actin. Two proteins are also associated with the strands of actin. These are globular troponin and two strands of tropomyosin. The troponin-tropomyosin actin complex conceals the binding site for actin. Now coming to organization of actin filaments. Individual actin filaments are assembled into two general types of structures called actin bundles and actin networks, which play different roles in the cell. In bundles, the actin filaments are cross-linked into closely packed parallel arrays. In networks, the actin filaments are cross-linked into orthogonal arrays that form three-dimensional meshworks with the properties of semi-solid gels. The proteins that cross-link actin filaments into bundles, called actin bundling proteins, are small rigid proteins that force the filaments to align closely with one another. The proteins that organize actin filaments into network are large flexible proteins that can cross-link perpendicular filaments. After discussing the structure and organization of actin filaments, we will now discuss about their assembly and disassembly. Assembled from asymmetric monomers, each actin monomer has tight binding sites that mediate head-to-tail interactions with two other actin monomers. So, actin monomers polymerize to form filaments. Assembled actin filaments have polarity, a barbed or plus end and a pointed or minus end. This polarity of actin filaments is important both in their assembly and establishing a specific direction of myosin movement relative to actin. Actin polymerization is completed in three steps. Nucleation. The first step in actin polymerization is the formation of a small aggregates consisting of three actin monomers. This step is the rate limiting step of actin polymerization. Two types of proteins, formin and the actin related protein complex ARP213 complex, determine where filaments are formed within the cell by nucleating actin filaments. Formins are a family of large 140 to 200 kilodalton barbed and tracking proteins, and both nucleate the initial actin monomers and then move along the growing filament, adding new monomers to the barbed end that make up stress fibers and the thin filaments of muscle cells. Many of these filaments are relatively stable due to filament stabilizing proteins such as members of the tropomyosin family. Tropomyosins are 30 to 36 kilodalton fibrous proteins that bind lengthwise along the groove of actin filaments. Elongation phase. Rapid polymerization of actin monomers onto previously formed oligomers. Actin filaments are then able to grow by the reversible addition of monomers to both ends, but one end, that is the barbed end, elongates 5 to 10 times faster than the pointed end. Steady state. Polymerization is limited by decreased monomer concentration. An equilibrium is reached between the filaments and actin monomers in solution. The rate at which actin monomers are incorporated into filaments is proportional to their concentration. So, there is a critical concentration of actin monomers at which the rate of their polymerization into filaments equals the rate of dissociation. At this critical concentration, monomers and filaments are in apparent equilibrium. The critical concentration is a measure of the ability of a solution of G-actin to polymerize. ATP enhances polymerization at the plus end. Potassium and magnesium ions are required for actin assembly. ATP binding and hydrolysis play a key role in regulating the assembly and dynamic behavior of actin filaments. Because actin polymerization is reversible, filaments can depolymerize by dissociation of actin subunits, allowing actin filaments to be broken down when necessary. ATP actin dissociates less readily than ADP actin. This results in a difference in the critical concentration of monomers needed for polymerization at the two ends. This difference can result in the phenomenon known as treadmilling, which illustrates the dynamic behavior of actin filaments. Treadmilling requires ATP, with ATP actin polymerizing at the barbed end of the filaments, while ADP actin dissociates from the pointed end. Treadmilling is important in the formation of cell processes and in cell movement. Several drugs useful in cell biology act by binding to actin and affecting its polymerization. For example, the cytoskeleton bind to the barbed end of the actin filaments and block their elongation. This results in changes in cell shape as well as inhibition of some types of cell movements. For example, cell division following mitosis, indicating that actin polymerization is required for these processes. Coming to our third objective, that is to discuss the association of actin filaments with the plasma membrane. The association of actin cytoskeleton with the plasma membrane determines cell structure and function. Actin filaments are highly concentrated at the periphery of the cell, where they form a three-dimensional network beneath the plasma membrane. This network of actin filaments and associated actin binding proteins determines cell shape and is involved in a variety of cell surface activities, including movement. Red blood cells or erythrocytes have proven particularly useful for studies of the association of actin filaments with plasma membrane. The major protein that provides the structural basis for the cortical cytoskeleton in erythrocytes is the actin binding protein spectrin. The actin cytoskeleton is similarly anchored to regions of cell-to-cell contact called adherens junctions. In sheets of epithelial cells, these junctions form a continuous belt-like structure called an adhesion belt around each cell, in which an underlying contractile bundle of actin filaments is linked to the plasma membrane. Contact between cells at adherens junctions is mediated by transmembrane proteins called cadherins. Coming to our last objective, that is to study the role of actin and myosin in cell movement. Actin filaments, often in association with myosin, are responsible for many types of cell movements. Myosin is the prototype of a molecular motor protein that converts chemical energy of ATP to mechanical energy, thus generating the force and movement. Muscle contraction provided the model for understanding actin-myosin interactions and the motor activity of myosin molecules. Interactions of actin and myosin are responsible not only for muscle contraction but also for a variety of movements of non-muscle cells including cell division. So, these interactions play a key role in cell biology. Actin cytoskeleton is also responsible for the crawling movements of the cells across a surface, which appears to be driven directly by actin polymerization as well as actin-myosin interactions. Muscle contraction. Muscle cells are highly specialized for a single task contraction, and it is this specialization in structure and function that has made muscle the prototype for studying movement at the cellular and molecular level. There are three distinct types of muscle cells in vertebrates: skeletal muscle, which is responsible for all voluntary movements; cardiac muscle, which pumps blood from the heart; smooth muscle, which is responsible for involuntary movements of organs such as the stomach, intestine, uterus, and blood vessels. In both skeletal and cardiac muscle, the contractile elements of the cytoskeleton are present in highly organized arrays that give rise to characteristic patterns of cross striations. Most of the cytoplasm consists of myofibrils, which are cylindrical bundles of two types of filaments: thick filaments of myosin about 15 nanometer in diameter and thin filaments of actin about 7 nanometer in diameter. Each myofibril is organized as a chain of contractile units called sarcomeres, which are responsible for the striated appearance of the skeletal and cardiac muscle. The sarcomeres consist of several distinct regions visible by electron microscopy, which provided critical insights into the mechanism of muscle contraction. The ends of each sarcomere are defined by the Z disc. Within each sarcomere, dark bands alternate with light bands called I bands for isotropic. These bands correspond to the presence or absence of myosin filaments. The I bands contain only thin actin filaments, whereas the A bands contain thick myosin filaments. The myosin and actin filaments overlap in peripheral regions of the A band, whereas a middle region called the H zone contains only myosin. Sliding filament model. The basis for understanding muscle contraction is the sliding filament model, first proposed in 1954 both by Andrew Huxley and Ralph Niedergerg and by Hugh Huxley and Gene Hansen. During muscle contraction, each sarcomere shortens, bringing the Z discs closer together. There is no change in the width of the A band, but both the I bands and the H zone almost completely disappear. These changes are explained by the actin and myosin filaments sliding past one another so that the actin filaments move into the A band and H zone. Muscle contraction thus results from an interaction between the actin and myosin filaments that generates their movement relative to one another. The molecular basis for this interaction is the binding of myosin to actin filaments, allowing myosin to function as a motor that drives filament sliding, formation of protrusions and cell movement. The movement of cells across a surface represents a basic form of cell locomotion employed by a wide variety of different kinds of cells. Examples include the crawling of amoebas, the migration of embryonic cells during development, and the spread of cancer cells during the metastasis of malignant tumors. All of these movements are based on local specializations and extensions of the plasma membrane driven by the dynamic properties of the actin cytoskeleton. Cell movement involves a coordinated cycle of movements which can be viewed in following stages. First, cells must develop an initial polarity via specialization of the plasma membrane or the cell cortex. Second, protrusions such as pseudopodia, lamellipodia or filopodia must be extended to establish a leading edge of the cell. Third, these extensions must then attach to the substratum across which the cell is moving. Finally, during cell migration, the trailing edge of the cell must dissociate from the substratum and retract into the cell body. In most cases, cells move in response to signals from other cells or the environment. For example, in wound healing, cells at the edge of a cut extend lamellipodia and move across the extracellular matrix or the underlying cells to cover the wound. Dear friends, this brings us to end the lecture on cytoskeleton and cell movement. Hope you have enjoyed. Have a nice day.